Hi everyone. Today we will discuss how babies are generated, how embryos develop, and how gender is determined. Okay, first of all, for this part, we will first discuss male reproductive organs and their functions related to spermatogenesis. And then we will discuss about the female reproductive organs and the oogenesis. And lastly, we'll discuss how fertilization happens. Okay, so this is an overview of male reproductive system. We learned that the male gamete is sperm, and the organ where gametes are produced are called gonads. Testes are male gonads that produce sperm and sex hormones such as androgens. Testes are formed in the abdominal cavity during embryonic development and descend into the scrotum before birth. Undescended testes, they can result in infertility because spermatogenesis requires relatively lower than body temperature. And then the ductal system refers to the structures that transport the sperm outside of the body. Okay, the accessory glands secrete seminal fluid to maintain sperm viability and motility. Seminal fluid is an integral part of the semen, okay, so which contains both sperm and the seminal fluid, with about 5% of sperm and about 95% seminal fluid. And then, of course, penis is the organ of sexual intercourse. Let's see these structures in the next slides. Okay, so this slide shows the anatomy of the male reproductive system and the functions of its components. I have highlighted some of the important, those are the key information. I hope you can remember here, okay? Let's follow the movement of sperm to introduce these structures. First of all, the sperm is produced in testes. Then it moves to this structure called the epididymis. Epididymis is a long-coiled tubule that rests on the backside of each testicle. Sperm complete the maturation in epididymis. Okay, the structure connecting to epididymis here is called the vas deferens. So this is a duct that transports sperm from the epididymis to the ejaculatory ducts in anticipation of ejaculation. All right. So then the accessory glands, such as this here called the seminal vesicle, it secretes fructose and also the prostaglandins that becomes part of the semen. And then following here and then goes to this is called the urethra. So remember, urethra is a dual purpose duct. This is because it serves as the channel for semen ejaculation and also for the urine excretion. This is a posterior view of the male reproductive tract. Notice that many of these structures are paired, means they have two units. And then also inside the testicle, it is filled with the structure of tubule called a seminiferous tubule. Okay, so this is the specific location for meiosis. Okay, now let's watch a movie of spermatogenesis to understand in detail how the sperm is developed.
Sperm develop in the seminiferous tubules of the testes. Primordial sperm cells, called spermatogonia, lie dormant in the outer compartment of the tubules from the late fetal period until puberty. At puberty, the spermatogonia undergo mitotic divisions, giving rise to genetically identical cells. The cells grow gradually into primary spermatocytes, preparing to divide into cells half their size. Each of these primary spermatocytes undergoes a first meiotic division, forming two smaller haploid cells known as secondary spermatocytes. These secondary spermatocytes divide once more, giving rise to four haploid cells called spermatids, containing exactly half of the father's genetic material. Through a process known as spermiogenesis, the spermatids change shape, transforming into mature sperm cells. The spermatid's nucleus condenses, the acrosome appears, and the tail begins to grow. Much of the cytoplasm is shed. As spermiogenesis completes, the sperm cells break free from the Sertoli cell lining of the tubules into the lumina. The sperm cells are then stored in the epididymis, where they functionally mature.
So this is basically a summary of the video you just watched. I will not go through this in details, but I want to mention that in this cross-section of seminiferous tubule, you can see all stage of the spermatogenesis. And the Sertoli cell here is working as a nurse cell and helps in the process of spermatogenesis by secreting hormones and supporting testicular fluid. Remember that the sperm generated in testes are not completely mature. The maturation occurs in where? Yes, epididymis, okay? And the location of the testes is specifically important, as the process of spermogenesis requires a lower temperature to produce viable sperm. Okay, we just talked about that a while ago. So that specifically is about 1 to 8 degrees lower than normal body temperature. And then testes produce about 200 to 300 million spermatozoa daily. However, only about half or 100 million of these becomes viable sperm. And then the bottom picture showing here is a mature sperm and it's all the components. The anterior half of the sperm head is called a acrosome. So it's a cap-like structure. This is derived from the Golgi apparatus. Acrosome contains many digestive enzymes that are helpful for the sperm cell to enter into an oocyte. And the middle piece of the sperm is packed with a lot of mitochondria. A sperm uses the energy in the middle piece to move. And then, of course, the tail of the sperm, act like a propeller and help the sperm to swim. Now let's take a look at the female reproductive system. Okay, the female gonad is ovary, where the oogenesis occurs. And the oviduct, uterus, and the vagina are also components of female reproductive structures. And let's take a look at these structures in the next slide. So this is the anatomy of the female reproductive system, and the functions of its components are included here as well. And see the structure very close to ovary here is named the oviduct. In mammal, it's also known as a fallopian tube. When eggs are released from the ovary, and then they travel along the oviduct and then these eggs will either be fertilized by sperm to become a zygote here, or degenerated in the body, okay, if it's not fertilized. So the location of fertilization is in the oviduct, okay? Just remember that it's important. And then zygote will travel into the uterus and implanted into the wall of uterus, and the fetus will grow inside the uterus, okay? And then let's take a look at a video of oogenesis before we go into some details.
The ovaries produce secondary oocytes, the female sex cells. During fetal development, stem cells, called oogonia, go through mitosis. Some of these cells develop into 46 chromosome primary oocytes. At the onset of female puberty, tens of thousands of primary oocytes each form part of a primordial follicle in the ovaries. Between menarche and menopause, each primordial follicle develops into a mature follicle and its primary oocyte completes meiosis I. Some primary oocytes become secondary oocytes, 23 chromosome cells that begin meiosis II, then pause. Each month, one secondary oocyte is released into the uterine tube. Meiosis II is completed when the oocyte is fertilized. The resulting ovum can develop into a zygote.
Summary slide of the video. Showing here is a cross-section of an ovary that contains follicles in various stages of development. I will not go into details, but have labeled these keywords with red color. Remember that the primary oocyte is in the primordial follicle. When the follicle becomes mature, then the meiosis I is complete and results in one polar body here and then secondary oocyte. Okay, so they both are ovulated when the mature follicle ruptures. After that, the follicles become a structure named a corpus luteum. Corpus luteum is important because it is responsible for the production of the hormone named progesterone during early pregnancy. As the title shows, the oocyte is the largest human cell and can even be seen with the naked eye. It measures about 0.1 millimeter in diameter. We learned this slide before. Here it is shown as a summary of this section and reminds you that oogenesis is a much slower process than spermatogenesis. Now we introduce the stage from fertilization to the baby's birth. The whole process includes the fertilization, implantation, and pregnancy. Fertilization is a fusion of oocyte and sperm. The fertilized egg is called a zygote. Okay, 3 days after fertilization, a normally developing embryo will contain about 6 to 10 cells. Then by the 5th or 6th day, the fertilized egg is known as a blastocyst. That is a rapid dividing ball of cells. Okay, blastocyst has many tooth structures, and the inner loop of the cells will become the embryo, also known as ICM. The outer group will become the cells that nourish and protect it, namely the trophoblast. Okay, we'll show this structure in pictures a little later. Remember here is fertilization occurs in oviduct, right? When blastocyst travels from oviduct to uterus, it needs to embed it into the endometrial lining of the uterus to grow. And then this process is called implantation. Once implantation is completed, the blastocyst continues grow and getting to pregnancy stage, which can be divided into 3 phases. This first or second and third trimester. Okay, each trimester lasts about 12 to 13 weeks. During the first trimester, the fetus organ started to form, and the organ maturation happens in the second trimester, and then a rapid fetus growth occurs in the third trimester. Now let's see some pictures for each stage. Fertilization happens in the oviduct. When oocytes travel from the ovary to the uterus, and then they encounter the sperm. In fertilization, many sperm surrounding the secondary oocyte, right? So they secrete enzymes that dissolve the outer barriers surrounding the oocyte. but only one sperm can enter the egg. Okay, the penetration stimulates the oocyte to begin meiosis too, showing in this step 1 in this picture. After completion of meiosis 2, how many polar bodies will be formed? Yes, after meiosis 2, the 3 polar bodies are formed. And then in step 3, the sperm tail degenerated. You don't see this tail anymore. And its nucleus enlarges and fuses with the oocyte nucleus. Okay, and finally, in the step number 4, a zygote has formed, and the fertilization is completed at this stage. All right. But how are ovulation and fertilization related to our everyday life? I have downloaded a picture showing the relationship of ovulation and fertility within a menstrual cycle. I think this will be useful for everyone. So on average, menstrual cycles last about 28 days in women. Some are longer, some are shorter. But if we consider the first day of menstrual cycle as day 1, then there's a good chance that ovulation appears, that happens on day 14. Now there are apps available that can roughly calculate the ovulation day when you record your menstrual dates. And ovulation lasts about 12 to 48 hours. But then 2 to 3 days before the ovulation is the most fertile days if you would like to have a baby, right? But after ovulation, there still have a couple of days that can be fertile too. Although the chance after 17 days is very little. Although the fertilization windows and menstrual cycles vary from person to person, if a woman has a very accurate cycles, it might be easier for her to manage fertilization or even contraception. Then you may have a question, why the dates before ovulation are the most fertile days showing here? There's no egg released during those days, right? So what do you think? Okay, so the key is that after ejaculation, sperm can live inside the female body for up to 5 days. The environment in a woman's reproductive tract is quite suitable for survival of the sperm. Additionally, remember that fertilization occurs in the oviduct, right? So the sperm cells must swim through the cervix and through the uterus to reach the oviduct for oocyte. This is actually a long journey for sperm cells to make, and a few sperm can survive. Okay, now let's see next stage, from fertilization through implantation. Okay, the zygote keeps dividing until the blastocyst forms, showing in this step. When the blastocyst travels to the uterine cavity, it starts the process of implantation to the surface of inner layer of uterus, which is named as endometrium. All right, showing here. The inner cell mass in the blastocyst here give rise to a disc-shaped early embryo. The trophoblast refers to the surface layer of the cells of the blastocyst. These cells provide nutrients to the embryo and develop into chorion and later a larger part of placenta. As the blastocyst implants into the uterus, cords of chorionic cells start to form. Okay, when implantation is complete, the blastocyst is buried complete in the endometrium. At this moment, the chorionic villi here showing the folds of tissue and blood vessels that connect the maternal and the fetal blast pools. And the yolk sac showing here is a small structure outside of the embryo. It is important in early embryonic blood supply and much of it is incorporated into the primordial gut during the 4th week of development. Implantation does not always succeed. There are many potential causes of errors. If implantation does not occur, the endometrium breaks down and sheds along with the blastocyst as part of the menstrual cycle. However, if a blastocyst does implant, then the endometrium remains in the uterus and together with uterine tissue becomes the maternal portion of the placenta. Now we get into the stage of fetal development. I will not go into details. The key concept to remember is the major events in 3 trimesters. Okay, for instance, this slide shows human embryo at 4 weeks after fertilization. It is in the 1st trimester, and you can see major organs started to form. This is embryo at 4 to 5 weeks of development. Organs continue to form. Heartbeat can be detected after week 5. By week 8, embryo completes the embryonic development period and started to transition to the fetal stage of development. Now it is already look like a human being, and the major organs start such as the heart, brain, pancreas, kidneys, liver, these are formed. Arms and legs are also well formed. And then the fetus continues to grow. In the 2nd trimester, your baby's features develop and organs become mature. You may be able to feel baby move. In the 3rd trimester, the baby will grow rapidly to get ready for birth. The fetus continues to grow. In the 2nd trimester, the fetus organs become mature. The mother may be able to feel the baby's movements. And then in the 3rd trimester, the fetus will grow rapidly to get ready for birth. Birth is a hormonal induced process. The hormone called oxytocin, released from the pituitary gland, causes uterine contractions during labor and helps shrink the uterus after delivery. Oxytocin is sometimes known as the cuddle hormone or the love hormone because it is also released when people snuggle up or bond socially. All right. So during labor, a cervical opening dilates in stages to allow passage of the fetus, and the uterine contractions expel the fetus. A short time after delivery, a 2nd round of uterine contractions begin and expel the placenta. Okay, the doctors need to check if the placenta is intact. Retain the placenta could cause heavy bleeding after labor. Why? Because placenta is directly connected to the mother's circulation system. Now let's see if environmental agents can harm embryonic development. Okay, teratogens are substances that may cause birth defects via a toxic effect on an embryo or fetus. Most common teratogens are listed in this slide, including X-rays, certain viruses, certain medications, and alcohol. Alcohol is known to act as a teratogen. Prenatal alcohol exposure, or PAE, remains the leading cause of birth defects and neural developmental abnormalities in the United States. So it affects 9 to 50 per 1,000 live births in the United States. Okay, now let's take a look at how these teratogens affect embryonic development. The effects of most teratogens begin after 3 weeks of development. Dark blue shown here represents the period of high sensitivity, and those light blues shown here, those are the period of developmental with less sensitivity to teratogens. From this chart, you can easily tell that the central nervous system is most sensitive to teratogens, and the effects can span through the entire pregnancy period. You can also see that teratogens cause major morphological abnormalities in the first 8 weeks. Why is that? This is because major organs form during the first 8 weeks. Any damage from teratogens in this period would interfere with organ development. I would like to focus on fetal alcohol syndrome. So this is a condition in a child that results from alcohol exposure during the mother's pregnancy. Fetal alcohol syndrome causes brain damage and growth problems. The problems caused by fetal alcohol syndrome vary from child to child, but defects caused by fetal alcohol syndrome are not reversible. Okay? So signs and symptoms of fetal alcohol syndrome may include any mix of physical defects, growth retardation, the facial abnormalities, intellectual disabilities, and learning disabilities. But how alcohol consumption in pregnant mother affected the fetus? This is because alcohol can enter mother's bloodstream and reach the fetus through placenta. The fetus metabolizes alcohol much slower than adult, and therefore can harm the development of tissues and organs and cause permanent brain damage in the baby. So never drink when you are pregnant. Now I'll move to our next topic, sex determination. Okay, we learned that the genome or genotype for female is the typically XX, but internal syndrome, we learned that only have one X chromosome, right? And the individual displays a female phenotype. In males, typically the genotype are X and Y, and in fact, any individuals with a Y are always a male. We will discuss why this is the case in a while, but you can imagine that the gene products, meaning the proteins made by genes on X or Y chromosomes, are important for sex determination. Okay, although environmental factors may contribute to sex determination, they have to interfere with the gene products. Okay, the effects of environment to sex determination is more obvious in certain lower species which do not have sex chromosomes. So for instance, in some reptiles that lack sex-determinant chromosomes, the temperature at which the fertilized egg is incubated determines the sex of the offspring. For alligators, the higher temperature produce more males, but this is the opposite in turtles. Then I can imagine that in Houston, we may have more male alligators than female alligators. In human, we have the XX and XY genetic system to determine the sex. Primary sex determination is the determination of the gonads. In mammals, primary sex determination is strictly chromosomal and is not usually influenced by the environment. The sex of an individual can be defined at several levels. The first is chromosomal sex, then the gonadal sex, and the phenotypic sex. Okay, the chromosomal sex is determined during the fertilization, but internal and external sex organs do not begin to develop until the 7th or 8th week of pregnancy. Before that, the external genitals of the embryo are neither male nor female. Okay, The gonadal sex differentiation is the process of forming ovaries or testes from undifferentiated gonads. It takes place over the next 4 to 6 weeks. If everything goes well, external reproductive structures can be established. Okay. In most cases, all these definitions are consistent, but things can go wrong during the development. I'll give you some examples soon. So now let's see how sex is determined during embryonic development. In the early embryo, shown here is in 6 weeks old of embryo, 2 duct systems. One is called a Wolffian duct, another is called a Mullerian duct. They are present at the same time. The Wolffian ducts are progenitors of upper male genital tract and give rise to the epididymis, vasa differentia, and the seminal vesicles and the ejaculatory duct. But then the Mullerian ducts are progenitors of the female reproductive tract. They differentiate to form the fallopian tubes, the oviduct, the uterus, and the cervix, and the superior aspect of the vagina. So which developmental ways to choose is really depend on the presence or absence of a Y chromosome and the gene called SRY. SRY is a gene located on Y chromosome, and it encodes the SRY protein. We know the central dogma, right? So you have a gene, you have a protein. So SRY protein is a transcription factor and the driver for development of testes from non-specific gonad. All right. So once testis development is initiated by action of the SRY gene, cells in testes secrete 2 hormones. One is testosterone. Another one is anti-Mullerian hormone, or AMH. So testosterone stimulates the Wolffian ducts to develop into the male duct system and external male structures. And the AMH, anti-Mullerian hormone, stimulates regression of the Mullerian ducts and prevents the development of a female reproductive system. So it drives the testes, but in the meantime, it will prevent the ovary development. So if there's no Y chromosome, what will happen? In fetus with XX, there's no Y chromosome, meaning there's no SRY gene. Then the non-specific gonad develop into ovaries in default, okay? Because ovary does not produce testosterone, and then the Wolffian duct system degenerates, okay? And because no anti-Mullerian hormone is produced, the Mullerian duct system develop into the female reproductive organs and structures. This is default. Okay, then this slide summarizes the major pathways of sexual differentiation and the stages at which genetic sex, the chromosomal sex, same thing, okay? Gonadal sex and the phenotypic sex are established. We have gone through those, so I will not repeat it. Now you should understand that why without a Y chromosome, the fetus show female phenotype. And with Y chromosome, the fetus show male phenotype. This is all because of SRY, right? Okay, in the pathway from chromosomal sex to phenotypical sex, many proteins are involved, and the mutation on certain proteins can disrupt the pathway and results in gonadal or phenotypic sex not matching the chromosomal sex. Okay, these events can occur 1 in 2,000 births. There are some examples. For instance, gonadal intersexuality. This can result from inefficient production of testosterone in the XY fetus. Without enough testosterone, the XY fetus can develop into a female phenotype. However, in very few cases, testosterone can be produced normally during puberty and stimulates male sexual development. Okay, another example is called a complete androgen insensitivity. Okay. Testosterone is a type of androgen and functions through binding and activating its receptor. This receptor protein is called the androgen receptor. If androgen receptor gene has mutation and loses its function, for example, if androgen receptor cannot bind to testosterone, then even in the presence of testosterone, cells of the embryo do not respond to their presence. As a result, development proceeds in the default pathway, which is what? Female, right? The female in picture is Sensi, so the woman in green shorts. So she's a phenotypic female, was reported to have an XY chromosomal condition and androgen insensitivity. She is an Indian track and field athlete and the winner of 12 international medals for India. Unfortunately, she was stripped of a silver medal won at the 2006 Asian Games after failing a sex verification test because chromosomal level, she has a X and a Y chromosome, so supposed to be male, but phenotypically she is a female. Okay, lastly, I would like to introduce the concept of X chromosome inactivation. We know that all females have 2 X chromosomes. Males only have 1 X chromosome. Then does it mean that females express more proteins from genes on X chromosomes? This is actually not the case. Interestingly, in female mammals, 1 copy of X chromosome is inactivated or silenced. Here, inactivation or silencing means no gene expression, okay? Because of this mechanism, the gene expression from X chromosome in females and males is equalized. So in 1961, an English geneticist, Mary Lyon, proposed this hypothesis to describe X chromosome inactivation. The hypothesis suggests, 1, only 1 X chromosome is active in each cell in female mammals. The other X chromosome is inactivated. Second, X inactivation is random, meaning the inactivated X chromosome can be paternal or maternal. So once the X chromosome is inactivated, it becomes a structure called a Barr body. Okay, let's see a short video on X inactivation.
X inactivation is a process that mammals use to equalize the gene dosage between males and females. Since normal male mammals have only 1 X chromosome and females have 2, there is a potential imbalance in the expression of genes on the X chromosome between males and females. In female mammals, this imbalance is prevented by inactivating all but 1 X chromosome in all somatic cells. X inactivation can be divided into 3 phases: initiation, spreading, and maintenance. Initiation occurs during embryonic development. Each X chromosome has an X inactivation center, or XIC. The number of XICs are counted, and 1 X chromosome is randomly chosen to remain active while the other is targeted for inactivation. Spreading is the process of compacting and inactivating the X chromosome. A gene designated XIST within the X inactivation center encodes an RNA that coats the X chromosome and promotes compaction. beginning at the XIC and progressing toward both ends until the entire X chromosome is inactivated. When spreading is finished, the compacted chromosome is called a bar body. After the bar body is formed, maintenance begins and continues through adult life. The bar body is replicated just like other chromosomes, but both copies of the replicated bar body remain highly compacted and essentially inactive. Thus, once an embryonic cell undergoes X inactivation, all cells that derive from it will have the same X chromosome inactivated.
In mammals, the XIST, we actually call the exist. So the exist gene encodes the exist RNA. Okay, this RNA does not make protein. Instead, it directly codes the inactivated X chromosome and helps to silence the chromosome. Therefore, exist is a marker for inactivated X chromosome. So this picture is the chromosome staining picture from a female mouse cell. So the expression of exist codes one X chromosome. So this showing as a red because the probe was labeled with a red fluorescent marker. So when you see this picture, you immediately know that this is a female cell, right? This is a picture of bar bodies, also the inactivated X chromosome. Showing here is a DAPI staining, only stands for the DNA inside the nucleus. Okay, so the A, this is a XY, the top one is the XY males. They have no inactive X chromosomes, no bar bodies. And then the picture here shows in XX females, they have one inactivated X chromosome and therefore one bar body, and showing those concentrated staining near the nuclear envelope. This is the bar body. And the C here is an abnormal female. This has 5 X chromosomes, okay? And then in this situation, 4 of those X chromosomes are inactivated. Therefore, you will be able to see 4 bar bodies. They are stained also as this blue color near the nuclear envelope, okay? So all the 4 X chromosomes are inactivated, only 1 left, which is activated. Because the X inactivation is random and can happen in paternal X chromosome or maternal X chromosome, there are 2 different cell types in female mammals. Some express alleles from the mother's X chromosome, and some cells express alleles from the father's X chromosome. This suggests that female mammals are actually mosaics, right? The calico cat is a good example. In this type of cat, an X-linked gene for coat color has 2 alleles. A dominant allele that produces orange colored fur, showing as the capital O here, or a recessive allele, or we'll show the lowercase o, that produced a black fur. Because X inactivation is random, the calico cat shows different colored patches of orange and black. Then you can tell from here that calico cat are always female. Same thing happens in human females. If a woman is heterozygous for a X-linked recessive order named anhydroectodermal dysplasia, she will also show mosaic pattern on her skin, not visually, but depending on which X chromosome is inactivated. If the X chromosome with dominant allele is inactivated, the recessive allele will not transcribe, and the affected skin will not develop normal sweat glands. So those are the showing the yellow color here, but not actually yellow, but just saying those are the affected skin. But if the recessive allele is silenced, then the part of the skin is normal, because this is basically you express this protein correctly, right? Okay, that's all for today. I'll see you next time.